again. So we will drop the last part of on information processing because we will not have time to get there. And the last part that will be now given by Francesco will be on circuit theory and metabolism. So this will be how to apply the theory that I will develop in the first 3 lectures, which is the basic formalism of thermodynamics of open chemical reaction networks. And maybe if people want, on the last days during the tutorials, Francesco can tell you a bit more about information processing. He has anyway prepared the lecture already. This is the stage that we reached yesterday. So we were considering, we opened the network by introducing fluxes and defined the dynamics for the open chemical reaction network. Then the argument was like, let's consider a closed chemical reaction networks as a mixture at equilibrium. We looked at the definition of the Gibbs free energy describing a mixture at equilibrium. And we got an explicit expression for it, taking into account the fact that we are dealing with ideal dilute solutions. And from the fact that equilibrium is a minimum, I argued that when we increment a little change of concentration due to a given reaction, the condition of the minimum requires that the delta G variation is 0, and as a result, that I could derive this condition related that I define as delta G 0, so the change of the mu zeros. Remember, mu zeros, think of them as the energy encoded inside the molecules. So this is really the energy released by a reaction rho. And it's that free energy, which contains both energy and entropy, that is stored inside the molecules. And that quantity connects to the ratio of the kinetic constants. It's highly non-trivial. We have a connection between thermodynamics and kinetics, and also the equilibrium of that reaction. Okay? And this is what I want to call local detail balance, this connection between the ratio of the kinetic rates and the energy changed during a given reaction. So, okay? So what I anticipated last time, so we are still in part 3, in part 1, big 1, B, thermodynamics. But this was the part equilibrium, and now we go out of equilibrium. And I will simply rewrite the form of the Gibbs free energy that we had at equilibrium, but assuming, without requiring now, that the concentrations are equilibrium concentration. I allow them to be any concentration. Okay? So this will be the definition of the Gibbs free energy. This is the dependence minus RT times the concentration of Z. Okay, yeah, this is not a vector anymore. So this is what we got yesterday, but with ZX, okay? And now I write it for any Z. And I'm saying this is my non-equilibrium Gibbs free energy. And we will rationalize this now. I already know that when Z is ZX, it is equilibrium. And to rationalize this, let me also kind of give the non-equilibrium analog of the delta G by the delta G 0, sorry, by defining the delta G now as the change of mu along a reaction. So that is now the full mu. It depends on concentration Z times the stoichiometric, the column of the stoichiometric matrix corresponding to the reaction rho. So this is the change of mu, which, remember, mu contains the mu zero part and the this contains the mu zero part and the RT log concentration part. Okay. So because of that, we can split this into the mu zero part that is corresponding to the delta G zero. And the remaining part that is the RT log of the concentration multiplied by the stoichiometric matrix. And so remember that this is a sum log of a vector. It's, you have to think of it component-wise. So I can transform this as RT log of, with the notation that I defined yesterday, Z to the power of the stoichiometric matrix with a minus sign. Why do I say with a minus sign? Ah, here. You're saying this is a minus? Wait, I need to get back on my feet now. This was plus, this was. Yeah, here there's a minus missing. And also here, but that doesn't affect my result, right? Because now I define delta G. This is plus, I think this is correct. Francesco, have you identified the map? Where? Here? Without the minus. Okay, but then I will miss a minus, I think. Right? Okay, let me do it. So the delta G I will replace by the kinetic constant, by this. Theoretical physics is all about minus signs and factor twos, as you should know. And then plus RT log concentration 2S rho. Ah, yes, and there I get my minus sign. And now this is minus RT log of K rho to the concentration of Z. And then I need to, this is where this, I always need to be careful about the signs because the numerator has to be it's in my definition of rho. You remember when I defined R, I splitted my fluxes into forward and backward fluxes. So this is mu rho. This is mu minus rho. And therefore, that ratio here is R rho over R minus rho. By definition, okay, this was the definition of R rho. This was the definition of R minus rho. And there's just one thing I want to make sure. Ah, yes, because I used the minus. Okay, I was confused. That's where my mistake comes from. Mu minus, see, this was by definition this, where this was product reactant. But because I put it together with the minus here, so I changed the order of the fraction. That's why I was confused. So this is correct. Okay, so now what do I show here? I show that the delta G, so the change of chemical potential, the full one, not the mu zero one, due to a reaction can be expressed in terms of, you see, minus RT log the flux forward in the reaction divided by the flux backward in the reaction. So this is the difference of flux is really measured by this delta G. Okay, and this will be very important for what I'm going to do next. Now what I'm going to do is take the derivative of G, and out of one of the term will be this delta G. So let's do it. So time derivative of G, this G here, this one. Okay, so what do we get? We get the derivative of these terms times this, plus the derivative of this multiplied by this term. So this is sum over alpha, mu alpha. Sorry, derivative of mu alpha over dt times z alpha plus sum over alpha mu alpha derivative of z alpha. Okay. Now what about this guy? The derivative of mu. So this is log, only the mu zero doesn't depend, where is my mu? Here. The mu zero doesn't depend on the concentration, so it's only the derivative of this. So it's RT, derivative of the log is 1 over Z. Okay? So 1 over Z times the derivative of Z alpha. But you see now that this goes away with this. Yes? Yes. I was about to put it. But now all this, because what is left here is this. I forgot the RT. So all this now goes away with the RT. right? Because what I'm left here is RT times the change of concentration. And here I have minus RT times the change of concentration. Good. So now the only term left is mu alpha times the derivative of the concentration, which I'm going to replace now by the stoichiometric matrix acting on the flux, because you remember this is our starting dynamical equation here, plus mu times I. And this guy here is my delta G. Delta G, yes. Delta G here is mu times S. Yes? There? Yes, correct. Thank you. Okay, now I'm going to use this expression that we got before for delta G, which is minus RT log R rho over R minus rho. And if you remember that J is rho, is R rho. Okay, now it's, how do I do that? It is a vector minus, plus, minus, minus. Okay, so these are the vector of the fluxes in the concentration. So this entire term here is has this specific form, which is typical of the entropy production. And indeed, I will define it as minus T, the entropy production rate, where the entropy production rate is going to be now defined as RT log, well, actually, usually it's written, let me simply reorder it, because it's more commonly seen like that. You agree? So I took out the T and the minus. So what is left is R times log of this ratio multiplied by the difference between forward and backward flux, which is the net flux. Which one is? Yeah, sorry, this is a minus rho. No, because I took the minus here. I defined this entire thing as minus T sigma. Therefore, okay? And now I call this the entropy production. This quantity is super important. Why? Because, and I forgot to sum over the reactions. Right, because this is a sum over the reaction, this dot product. I've taken it out. Ah, yes, sorry. Okay, why is this super important? Because this measures the asymmetry in the net flux. This is the net flux. So look, if this is positive, is the forward is bigger than the backward. This is bigger than that. So this is bigger than 1. and the log of something bigger than 1 is positive, so this is positive. If this is smaller than this is negative. That ratio is lower than 1. The log of something lower than 1 is negative. So the full thing is again positive. This quantity is always greater or equal than 0. And furthermore, not only the sum, this is true for each of the terms. This is also very important. So each of the terms are separately positive. And they are only 0 if the forward and the backward flux vanishes. And remember, this is exactly. So sigma dot is 0 only at equilibrium. So this quantity, equilibrium, I cannot speak well, right? So this quantity measures how far away we are from equilibrium. It's a metric. It's really had the structure of a relative entropy. You don't need to understand the details of that. This is a metric from information theory. So it measures really how different the forward flux is from the backward flux. Okay, now it also shows us this statement. I want to go back to the delta G now, because up to minus T, this is the delta G of the reaction, right? So this also shows me that the delta G of every reaction is positive. So in other words, that the delta G of every reaction has to be negative. Because delta G, yeah, but remember that delta G up to a factor minus T is the entropy production. So the positivity of each term into that sum implies the positivity of the delta G of the negativity of the delta G of every reaction. So very important result. Every elementary reaction can only flow down in free energy. This, you might say, okay, but what about energy transduction? Because we discussed that energy transduction is going up. So we will see that energy transduction never happens at the level of elementary reaction. It only happens at the level of effective reactions. And this will become very clear when we do circuit theory. The reaction. There's no trajectory here. The reaction. See, this is the delta G. It's here. Let's go back here. The delta G is the change of mu due to a reaction. So at the most, it's per mole usually because we deal with concentration. But you can think of it in terms of molecule. One increment of a reaction changes the energy of the reactant end product by a certain amount. And that amount can only go down. Okay, now we can rewrite this equation because I haven't spoken about this term yet. This term, I'm a bit messy, I have to say, with this square here. So I define this as the chemical work. Why? The rate, because it's per unit time. This is the amount of free energy, chemical potential, that I bring in to the system due to the exchange species. So this is really what is brought inside the network by this exchange mechanism, right? I is the vector, is only non-zero for those species that are exchanged. So I times mu is the energy I'm bringing into my system from the outside. And that's why I call it work. And so with that, I get the second law, basically, in what I would call a Kelvin-like form. You see? entropy production is work minus free energy. For those who are familiar with the formulation of the second law in thermodynamics, this is usually the formulation which involves work and free energy is sometimes called the Kelvin formulation. We will then also show the Clausius form in terms of heat and entropy. Okay, one thing that we can already say is that in a closed CRN, the chemical work by definition is zero because it's closed, right? We don't bring any free energy. So what can we say about G? It's always going to go down. If you don't supply energy to your system, G is always going to go down. Dot. Well, yeah, dot. Both actually. Even if I integrate it, it's zero. If I integrate it over time. Because this term is only non-zero if the flux is bringing stuff in. So as soon as I close my system, that term becomes zero. I will show you later, because for that I need a bit of topology, so I will not prove it yet, but one can show that J is always greater or equal than Jx. Proof later. So this also shows you that this thing can only go down in a closed system, because if you don't supply work, it's going down, until it's going to hit equilibrium. So a closed system always relaxes to equilibrium, and J always goes to Jx. If you want J to go up, you need to supply energy via chemical work. Good?
I have one question.
Yes.
Here you showed that the entropy production rate has to be greater than zero. The
Yes.
argument was by multiplication of the R and log terms.
Yes.
But then you took this argument to delta G less than zero. But delta G and entropy production has a difference, right? There is no J term there, the flux term. So will the argument follow directly?
No, no, you're absolutely right. It's, I need to multiply by the flux. Yeah. So, of course. So, yeah, you're actually absolutely right. Thank you for pointing this out. So the point here is that, thank you, important, is that the flux of the reaction, I was verbally implicitly taking this into account, but it was misleading because I hadn't written it down. It means that the flux is always going to go in the direction of minus delta G, right? So if delta G is negative, my reaction goes forward. If delta G is positive, my reaction goes backward. Yeah? Because of the negative sign here. So the delta G, if you take, this is something in textbooks, actually one of in the previous talk, there was the list of the delta G for glycolysis. They sometimes plot this delta G using physiological conditions, because this delta G contains a concentration here. So you can plug the physiological conditions for every step of glycolysis and see how far from equilibrium that reaction is by looking at delta G. And there's something intriguing which I still don't fully understand, is that the most regulated step are the one with the biggest negative delta Gs. This is I think puzzle, I mean, interesting observation that, I mean, one could say, yeah, because regulation requires to be out of equilibrium, but this is very vague. I think this is an interesting observation that was made already in these textbooks. Okay, yes. Thank you for pointing this out. So the steps in the glycolysis which are known to be the most regulated ones are the ones which have a large delta G. So that go very strongly downstream. While the other steps have small delta Gs, they are quite close to zero. So it's like, you know, some steps seem to be close to equilibrium, but those steps that are regulated seems to be the ones far from equilibrium. Yes. Yes. I'm anticipating a bit here. This is a side remark. Good. So we have the second law. That's already a good thing. Now let's go to the first and second law, but the other second law in the Clausius form. And for that, we need to remember that in thermodynamics, the mu zero can be split into an enthalpic part and an entropic part. Okay? I think this is, I can assume that this is more or less known, right? So mu zero has an enthalpic part and an entropic part. So if I plug this now in my definition of J, that is here, what I can do is the following. I can write J. I write it and then we verify it. Is going to be the sum of what I define as big H and big S, the enthalpy and the entropy of the entire chemical reaction networks, where yeah, let me write it. Where, so let me start by H. It's the simplest one. H is simply the sum over the H0 times the concentration of Z alpha. And S is the sum over alpha of the S alpha 0 plus RT log Z alpha minus RT, everything multiplied by Z alpha. Okay, enthalpy, so let me write it, enthalpy. And enthalpy, keep in mind, as I said, pressure and volume do not change here because we are essentially in water. So you can really think of enthalpy as energy, because enthalpy is energy plus PV, but any change of PV here will be zero. So we are essentially speaking of energy when we are talking of enthalpy, because we are in dilute solution. And this is really the entropy of the chemical reaction network. So you see that what I did is, this has the mu zero part, plus the RT. Where is it now? Here, no. I just need to stick things on my blackboard. Where did I put mu? There. So there's the mu zero plus RT log Z and the minus RT. So I split the mu zero in enthalpy and entropy. The mu zero, the H0, the enthalpic part, I simply keep very simple as the expectation value kind of the enthalpy. And all the rest I put in the entropy. And entropy has non-trivial fact. It has this log Z, a bit reminiscent of a Shannon-like entropy, but now we are there, don't forget that we are speaking of concentrations. These are not probabilities. The sum of the Z's are not normalized. So it's not really a Shannon, it's a kind of generalization of Shannon. And this RT term is also in the entropy. And now if we take the balance, No, here I don't think. Why? Because this is really the log of the concentration times the concentration. Because mu, remember, here, mu has the mu0 part, which I now split into enthalpic part minus entropic part. But the RT log Z is the one that ends up there. Okay, here. And then the RT factor. Yes, but I'm multiplying because big J contains also you can think of it as a kind of average. It's this guy here. Right? It was mu minus RT times Z. So the Z outside is this one. Okay, so now let's take derivatives of the enthalpy and the entropy. And as I said, since enthalpy is kind of the energy, this is going to be our first law. So this is going to be, now I simply take this one is trivial, right? Because it acts only on Z. So it's going to be H times, so vector, matrix, vector, plus H times I. And now, yes.
Massimiliano,
Yes.
for the entropy term, the T terms should not be there. For the capital S.
Thank you very much. Good, I have a careful audience. Indeed, because it is here. Yes? Indeed. Because of the minus here. Good. Any other corrections? Okay, first law. Now, this is known to be the heat. The enthalpy change during a reaction is the heat. So this is Q dot. That's really the heat that gets released in the solution when the reaction happens. And this is the enthalpy supplied from the outside. This is also 0. By the exchange fluxes. Let's now look at, again, the second law, but that now is going to be of a Clausius type. Or Clausius form, I call the previous Kelvin form. So here we have, it's a bit similar to before. So we have the derivative of these terms times this. So sum over alpha. The only term that depends on time is this one. So it's going to be minus R 1 over Z times derivative of Z. But times Z alpha. So this goes with this. And the second term is the sum over alpha of S alpha minus R log Z alpha plus R times the derivative of Z over T. So yes, again, this term here, the R times the derivative of the concentration, I cross it like this, goes away with this one. And I'm going to now define, this is the S0. Yeah. I'm now going to define the S alpha as S alpha 0 minus R log Z alpha. This is a definition. I incorporate the logarithmic part into the S alpha so that now this I can write as S vector times stoichiometric matrix times flux plus SY times IY. So this again is the entropy supplied by the surrounding. This is the entropy consumed by every reaction containing this log Z term. And since I can rewrite the S as enthalpy minus chemical potential, this is H0 minus mu, because where did I write it? That here. Now I'm using this with the RT. So this is going to be H0 over T minus mu 0 over T. And minus mu, because I incorporate now the RT log Z inside the mu. Yes. Oh, I'm going too fast. Let's do it step by step. Mu 0 minus R log Z. Yes, vector. Yeah, this is, okay, this is correct. And now all this, because this with this is the no. All this is minus mu over T, like this. So in summary, what I have now is H0 over T. Let me do this directly. So H0 times this, you remember, is heat, but heat divided by T. So the H0 part is going to be Q dot over T. The mu part times Sj is the delta J. Where is delta J? All the way there. Over T times J plus S times this. And remember now, this is entropy production. There. Because the delta G is minus T times entropy production. So overall, if you're still, if everything is not too ugly, it would be nice maybe, okay. Yeah, let me write as a summary here. equals, and I rewrite now this equation here so that it's clear. I get that ds over dt is equal to heat flow divided by temperature plus entropy production plus SYI. Okay. SI, which is equivalent to restrict on Y, because what this vector is only non-zero on the exchange species. Yeah. Is it clear? Maybe let me do the following. I simply rewrite what I just wrote here. Q dot over T plus entropy production plus SYIY. This is this. And now here I simply reordered the equation to make it look more second law-like. So this means that entropy production is dSET minus entropy reduction is ds dt minus q over t minus SYIY. Why is this more intuitive? Because you now, let me also emphasize that this is always positive, because you can say that this is the entropy change in the system. In the CRN. And this is the entropy exchange with the reservoirs. Two types of reservoir. This is the heat reservoir, which is the solution at temperature T. I'm releasing energy, but I'm isothermal. And this is the entropy exchange with the chemostats, with this exchange species, the objects that supply me the exchange species. Okay. Now I remember a comment of Francesco, who asked me to define the entropy flow. Let me do it like this, because it's kind of two perspectives actually to interpret the second law, and both are equally valid. So as I said here, this is the entropy change with the reservoirs. But there's also this perspective, which is, if ordered in this way, it's the change in entropy in my system is due to, it's a bit ordered in the wrong way. If you allow me to simply swap these two, okay? I first write this one and then this one, because I want to say that all this is something. So this is the entropy produced by all the internal processes, all the reactions going on in my system, and the entropy exchanged with the environment. And this is why sometimes this is defined, this is called the entropy flow. And you simply need to keep in mind that the entropy flow is minus the entropy change in the reservoir. So you can get both perspectives equally valid. This is the perspective where entropy production, the meaning of it is really the entropy change in the universe, the thing that always needs to be positive, because it's the sum of the change in the system plus the change in the reservoir. This perspective is more in terms of the change in the entropy is due to internal processes, which is sigma, and that one is always irreversible, and exchange processes that is called entropy flow. In Prigoghine's literature, usually this one is the most common. And
in the second interpretation,
yes,
what was Q dot by capital T?
the entropy change in, so I said reservoir here. So this is the
not that one, the other one.
Okay, let me write. I know I cannot write here because apparently the lights, nobody can see. So this is the heat reservoir. This is the exchange processes, those yellow stuff that are providing stuff from the outside to my system.
Yeah, in the other interpretation,
there.
in the other interpretation, what is Q dot by T?
It's how the energy flows out of the system. I mean, they are equivalent. It's simply a way to rationalize things. And this, I would say, is the Clausius-like version of the second law. Did I say that? Yes, I say that. Okay. So this kind of concludes the thermodynamics of open chemical reaction networks without topology. Okay, and these are really balance equations, if you want to think of them that way. We understood what defines the energy and the entropy of the system. We know that it's the free energy that is a combination of both that is relevant, and that gets minimized by the dynamics. This is nice, right? This is why we are really doing non-equilibrium thermodynamics here, because the system dynamically is gonna minimize the free energy. In classical thermodynamics, you only know that the system, the equilibrium is the minimum. You don't know anything about how you get there. So this shows also how dynamics and thermodynamics come together. And the key, this is why this thing is so important, local detail balance. It connects thermodynamics with kinetic rates. And that's what we call a thermodynamic consistent description, because we could, of course, solve the full dynamics, but this is complicated dynamics, and this is nonlinear dynamics. Not easy. But thermodynamics constrains these dynamics. These dynamics cannot do anything. They need to satisfy these conditions. If we close the system needs to relax to equilibrium. If we want to go up in G, in J, G, we need to supply work. Okay? Good. Interaction affecting pressure. Yeah, pressure, pressure, I've never thought really about it, because usually water is not so compressive. Temperature, yes. I mean, temperature could be relevant. Here we are assuming that the heat release in the solution is not changing the temperature, because water has a large heat capacity, and so to a first order. But I think an interesting challenge is to go non-isothermal, and because understanding why cells or living system, at least some living system, can maintain temperature constant is, I think, one of the very interesting challenge. And so this is something we would like to do, extend to non-isothermal, but we haven't done yet. Yes. Yeah. So this is important. It's sometimes called local equilibrium. So what we're saying there is that you can really picture reactions as reaction is the thing driving the system away from equilibrium. And what reaction do is, before the reaction, there's a solution at equilibrium. Now reaction takes a set of molecules, transforming it in another set. And you are assigning the energy to the product, which is again the equilibrium mixture energy. So there's a kind of non-equilibrium process that is the reactions connecting equilibrium states, which is the solution. The solution is always at equilibrium. And this has to do with, it's a kind of time scale separation argument. You're assuming that reactions are very localized in time and in space, but very quickly, the molecules re-equilibrate with the solution, so that next time a reaction occurs, everything is equilibrated from the solution point of view. You can view it as a kind of, the frequency of reaction is such that it leaves time for molecules to re-equilibrate after every reaction has occurred. That's the underlying assumption, which, by the way, in spirit is very similar to stochastic thermodynamics. When we identify states, we need to assume that those states have an energy or a free energy that is at equilibrium, and it's similar. So we need to identify, we need to capture all the processes that drive the system out of equilibrium, and these are reactions. And all the rest is assumed to be at equilibrium. We could go beyond. I think this is not too difficult within linear response for heat transfer, diffusion. Actually, we generalize to diffusion because all these things are reasonable. It is reasonable to assume Fourier law or Fick law. So you can linearize these fluxes into the forces. I'm getting a bit technical here, but just to, it's an important comment. The point is chemistry cannot be linearized in the sense that the chemical gradients are always very strong, but the other processes like heat transfer, matter transfer, they can be linearized. So actually they are the easy part. Generalizing this formalism to couple it to heat transport and particle transport is actually quite standard. You can use the standard recipe. The most important part is to capture the chemistry really far from equilibrium, and that does that. It's a bit cryptic what I said, given what I explained, but I can explain more of that if some of you are interested.
So
yes.
The question. So we are here assuming that all these reactions are irreversible, right?
Irreversible?
Yeah.
No, why?
Because like if we look at this term where we are taking time derivative of the Gibbs free energy, the time is like present in the equation in a linear sense. So like it will enter the equation linearly. So if we change the variable from t to let's say minus t, the equation equation won't remain invariant, right?
Yeah, in that sense, yes.
So it's irreversible.
Yes, chemical kinetics is irreversible. Yeah, that's why there's a second law. Yeah, we are not Newtonian. Yes. Good, now I will start. Did I mention this? Yes. Huh? Okay. So now I will start the slightly more difficult part. Is using topology. I don't think we'll get to the point of the second law using topology, but that's my goal. I want to show you that the topology of the network really is important to further split the expression of the entropy production that we have and identifying the forces, the thermodynamic forces that are driving the system away from equilibrium. So thermodynamic forces, if you're not familiar with that term, think of it as which are the relevant temperature gradients and chemical potential gradients that drive my open system away from equilibrium. Yes.
So the separation of timescale you mentioned is the conductivity, heat conduction, and the rate of reactions.
This is more, I don't even need to be specific there. It's really the fact that, okay, once the molecule collide, of course, they go very out of equilibrium during the collision, right? So entire process of the quantum mechanical process of reorganizing the bonds. Then they leave. As they go in the solution again, they start colliding with the solvent right after the reaction. They're gonna thermalize and reach again an equilibrium that is given by the mu0. Because I said this is a free energy of the molecule at equilibrium in the solution. And I'm assuming that they have time to get there before the next reaction kicks in. Part 2, using topology. And identify forces and non-equilibrium. Yes. Yes. No, because this is pure dynamics, right? There's no thermodynamics here. When you write this, the only thing that you know is that the reactions are balanced mass-wise, but you don't know anything about energetics.
This is actually telling you that it's well stirred.
Yes, this is true.
That's exactly what you're saying.
It's one aspect of what I'm saying, but the generalization to reaction diffusion is very easy because you simply make this space dependent and you add a diffusion term to the differential equation. So it becomes a field.
Assumption that you mentioned that after a reaction, things equilibrate with that is already assumed.
No, I wouldn't say, because here, when you do dynamics, you don't need to care about equilibrium a priori, right? Here, you're not even assuming mass action kinetics at this level.
But you're assuming well stirred.
Yes, I am assuming that because I'm describing my system in terms of only concentrations that do not know about space, that there's well stirred. But well stirred, I don't need to say anything about equilibrium. Equilibrium is much more, requires much more care to be defined. It's because I want to define energy. I want to assign an energy to these Z's that I need to start arguing as I did, right? If you do kinetics, you might have an effective kinetics in many, many cases, even if it's very hard to justify that the energetics is right. So this is, I want to emphasize this, that you can, it's much, I mean, I wouldn't say much easier, but when you write a dynamical equation, very often people don't care about energy. It's whether or not your system follows these dynamics, but whether or not what is at equilibrium, what is not, you don't care. I do care because I need to assign an energy to those dynamical variables. And then if I need to do it, I agree with you. So because I want to do it now, I need to assume well stirred because my starting point is equilibrium in the entire system as a uniform mixture. But it's easy to generalize to say, okay, I do local in the sense of space now, equilibrium. I assume that in every little volume, I am at equilibrium and it's a mixture. But there's diffusion that allows things to be transferred in space. And reactions are local. So the extension to add diffusion is really an easy one. And it remains within local equilibrium approximation because in every little volume, you're saying that reaction happen, the volume is mesoscopic, reactions happen, particle equilibrate at the temperature of that little box, and then you go on like that. So adding diffusion is conceptually not very different. Even adding heat transfers should not be a major issue. Simply the kinetic rates now change due to temperature, so this needs to be taken into account. But conceptually, I don't see major obstacles. And this time scale separation is usually pretty good, except if you would really go dense, very dense. But local equilibrium, you know, it's used to describe explosions. So it's even I was surprised to, but this is known that if people who study explosions, if you're interested of the wave fronts of the explosion, they use also local equilibrium. So, you know, if you want to break local equilibrium in a sufficiently large system, you really need to go for it. Okay, topology. There's a bit of linear algebra here. Huh? Don't be scared. So we'll start with definitions. Internal cycles. And all these terminology will make sense eventually. These are the vector of the right null space. So I stands for internal. So this is the definition of the null space of this matrix in linear algebra. And the number of independent vectors that span this null space is given by the number of reactions, because this matrix acts on vectors that have the dimension of reaction, minus the rank of the stoichiometric matrix. And the rank is the number of independent row vector, equivalently, because it's equal, also the number of independent column vector, row, column. This is linear algebra. So this, you know, you can, yeah, Simone? Yes.
Sample, maybe that could be useful.
Yeah, I will give them in a second. The problem of that, I told you, this example has been carefully chosen to illustrate almost everything except having an internal cycle. So you picked exactly the only thing. So that, the stoichiometric matrix of that object, which by the way, I should write because for the application. Let me quickly rewrite it. -1, 0, 1, 0, 0, 1, -1, 0, 0, -1, 0, 1, -1, 0, 1, 0, 0, 0, 1, -1, 0, 0, 0, 0, 1. Francesco, you check? Done. Here we go. That was our stoichiometric matrix. And that matrix happened not to have any internal cycle. Okay? So if you try to find a vector such that if you act with this matrix on that vector and you get 0, there's none, except 0. Okay.
Maybe you can show that the rank is 5.
Yeah, well, then you need to, that the what? Ah, yes, that, well, you need to show then that, exactly. But this is because you cannot take linear combinations to eliminate, all these vectors are independent from each other. So because what you need, the null space is if you find some that are linearly dependent. But let's the next step will illustrate this notion of cycle, because now I'm going to say that there's a typo in the second row. What is it? Thank you. So let me now define cycles as same thing, but now for the X part. You remember the X part is this part of the stoichiometric matrix. So these are and now the number of these cycles is still equal to this minus the rank of Sx. So there are two ways to see now that this is bigger than this. One is algebraic, because NR is the same, because as we cut the rows, we don't change the number of reactions. No, what I meant, yes, we don't change the number of reactions. Yes. And we may decrease the rank, right? So because we decrease the rank when we cut the matrix, we get that this is going to be bigger than that. And so we're going to call now the emergent cycles. Sorry, I said there's the algebraic way, and then there's the, and you can see it intuitively like that. Those, remember, are cycles of the full matrix. There is none, but if there was, it means that every time I do this product, right, and I sum this, for all of them, I get 0. But now, once I remove this, those that were, if I had an internal cycle, it would still be an internal cycle because all those rows are still 0, right? So I know that the old internal cycle will still be cycle of X, but I may get new ones. And the new ones are called emergent cycles. And those, so I will define with the label epsilon, and they are simply defined as the set of all the cycle contains the set of internal cycle and the set of emergent cycles. And now let's give an example, because this matrix has cycles, and they are by definition emergent cycle, because, I mean, not by definition, because I said there are no internal cycles. All the cycles will be emergent cycles, okay, because we have none of these. So all the cycles are emergent cycles. So let's check it. It's so one that we will call CW. It's the vector 1, 1, 1, 0, 0. And the second one is, I label by P, 0, minus 1, 0, 1, 1. Because now if we multiply, you can do it. So minus 1 times 1, this is minus 1, then 0, 1, 0, 0. So it's 1 minus 1, 0. And so you can try to, so these are the cycles, okay? Take these two guys here. First this one, then this one, but write it here and check that this is 0. Okay? You can try maybe this one, okay? Let's go with the, let's say the last row, this one. So in principle, it has to be true for all of them to be the case, but 0 times 0, 0 times minus 1, 0, 0 times 0, 0, 1 times 1, 1, 1 times minus 1, minus 1, so it's 0. So whatever you take as row and you multiply by these vectors, you get 0. So, yes. Now, one thing that, why are they useful? One way to already anticipate, they are sequence of reactions. You can read them like that. They are a sequence of reactions that leave the internal species unchanged. Actually, those cycles, because here it was for the full matrix, It's a sequence of reaction that would leave the entire system unchanged because there are only internal species, but I can not show it because there are no internal cycles. But let's look at the X matrix. These are sequence of reactions that do not change the abundance of the internal species, but because they are emergent, they change the abundance of the exchange species. So let's check that. Let's take this one. So 1 is I implement the first reaction. I have a 1 again for the second reaction, and a 1 again for the third, and 0 and 0 for 4 and 5. So what I'm doing is zack, zack, zack. What did I do? Look at the internal species. I'm back to where I started. But I took in some F, and I spit out some W. Let's look at the other one. So this is nothing of the first reaction. So -1 for the second. So I'm going this way, -1. And then 4th and 5, 4, 5. So this is chuck, chuck, chuck. I don't change the internal things. I'm back to where I was from the internal point of view. But from the exchange species point of view, I spit out this, and I get and I spit out this. Okay? Did I screw up a sign of the arrow? Ah, right. Yeah, I noticed that there was something wrong. Thank you. Yes, okay, good. So this is the meaning that these cycles have. And let's look at steady state. So remember that now we had these equations of motion for x that was Sx j. And let me look at steady state. So steady state, by definition, is this. Right? I don't want the species to change anymore. So what is this telling me? That this vector of steady state, because it's steady state, I put an SS here to emphasize that the flux is at steady state. I can decompose this steady state flux in terms of emergent cycles. It's a basis. They must live in the right null kernel of my matrix. So I can always express a steady state flux in terms of all the cycles, which I can decompose into the internal ones and the emergent ones. And now if I substitute this into the second equation, what I get is that Sy acting on this cycle. So the internal cycles were cycles of the full S, so it's going to be 0 when Si acts on them. So if I act now, if I substitute this here, what do I get? I get that Sy C epsilon multiplied by these epsilon things equals minus Iy. And this, you can think of this as the stoichiometry of an effective reaction. Let's see that. Let's write it for the two cycles that we had. So let's apply Sy. Remember, Sy is this lower part. Huh? It's this. Let's apply Sy on this guy. Let me do it here. Sy on Cw. So please check. I write them and you check. This is one result. This is the other result. Okay. So I need to. They're a bit far away from each other, but yeah, 0, 0, 0. Then I get a 1. So yeah, you can convince yourself. I'm doing it too again. Who says something wrong? No, it should work. So apply this to this, you get these two vectors. And now, since, remember now, this is a sum over reaction, so we are left with species, and these species are the species, the Y species. So just to remind you the order that we had, this was P, W, S, F. I didn't rewrote this here, but this is the guys that were here, okay? F, W, F, W, S, F, okay? So now this means that this effective reaction, what does it do? It consumes one F and converts it in W. And this one consumes one S and transforms it into P. Okay? And this, now remember what we said, we had that cycle, which was, spitting this out, taking this in, the other taking this in, spitting this out. These effective reactions do exactly that. So now the meaning of these guys is really, since from the internal point of view, nothing has changed when you go through a cycle, the effective reaction is the net balance of what happens from the external point of view, from the exchange species, once you complete the cycle. Can you please repeat?
Why do you have indices W and P?
Because of what they carry, because W and P are carried by the cycles. But this, in principle, there's a basis independence. So I could take linear combination of those and generate other ones. It's for convenience that we took those. And you will see also later why I chose this labeling. But take this as at the moment as an arbitrary labeling. Okay, so you got a bit of a sense of why these cycles, we can already anticipate that they are meaningful because they don't do anything on the internal system, but they do stuff on the external one. And we have this notion of effective reaction that appears when we apply SY on these cycles. Okay, and the next step will be conservation laws. I have no more time left, but the conservation law will be the left null vectors. So now we considered the right null vectors. Now we will look at the left null vectors. And both of these things are important. This is related to conservation law, quantities that are conserved, and that when we open the system are not conserved anymore. That will also be important. Yes. Because these ones, you have to keep in mind that I can do this at steady state. So if you're not at steady state, you may have accumulations also in the system. That's kind of an intuition. But these are not, conserved quantity are, you will see next time. These are expressed in terms of concentrations of various species. combinations of various species that are conserved. So they are really abundances. This is more, the cycle is a sequence of reactions. So in that sense, they are not conserved quantities. They are processes that if you go through them, they don't change the internal state, they only change the external. But this will be really, really important for thermodynamics later on, because thermodynamics really cares about the cycles and the conservation laws. More to come.
So it seems that you didn't really use the first equation, the dx dt. Is it because you don't have internal cycle?
No, I use it here to prove you, to justify that I can decompose the steady state flux in cycles. Right? Because I decompose J in cycles. And this I can only do because this is 0.
Okay.
Otherwise, I could not do that.
Okay.
What would an internal cycle look like? Could you give an example? Or,
Can you speak louder?
what would an internal cycle look like?
Do you have examples at hand? Yeah, right. You know a stoichiometric matrix by heart? Oh. I didn't define complexes, keep that in mind.
First reaction, second reaction, third reaction, fourth reaction. You get +1 for the first reaction, +1 for the third, -1 for the second, and -1. That's it.
And now nothing has changed. But since the system is closed here, by definition of a cycle, nothing has changed. While when you open, the cycle from the perspective of the internal species doesn't change anything, but from the perspective of the outside species, of the exchange species, it does change. You will have Francesco in the exercise session will give you a bunch of networks where you'll have to define the cycles, the conservation laws.
Look like a cycle. Like it's...
Well, you can draw it. Look, let's draw it as a hypergraph. Why is it so hard?
Okay, depends.
Okay, so 1, this is 2. This is 3, and this is 4. And so if we do, this was +1, -3. Okay, so the convention here is opposite. You took 3 like this, right? No. Okay, let's define the convention, otherwise we will also mess up. The first one is plus, like this. The third one is plus, like this. The fourth one is plus, like this, so we will go minus. And the second one, this was also plus, like this. Okay, so if we go, a cycle would be +1. So for the first reaction, +1. The third one, a 1. The fourth one, a -1. And this one, a -1. And the point is that, you see, look at what you do from the S and P point of view. Let's start here. I take in a S. I spit a P. I take in a P. So P and P now, one I spit out, one I take out. So I didn't do anything in terms of P. And now I get in a P. So S. And so I don't change S. So here nothing has changed. Yes?
Oh, so I just want to check if I understand. So is it correct to say that the balance of the products is forced out of equilibrium only when there's a flow of energy or matter into the system?
No, because you could take an initial condition that is not equilibrium in a closed system.
But if it starts in equilibrium, it's forced out only if there's a flow of energy or matter? Okay. And then if the products are out of equilibrium, is it possible that this disequilibrium then drives further reactions that creates enzymes or proteins?
This I didn't get.
So say there is a disequilibrium in the products. Is it that disequilibrium that can drive certain reactions?
Yes, any disequilibrium, but that's what I'm saying. It's the initial condition. If you start with abundances of species that do not correspond to equilibrium, the delta G's are non-zero. They are not there anymore. The delta G's are not zero anymore. And so the fluxes will kick in to re-equilibrate every reaction in the closed system. Huh? In the open, it's different because then you start supplying, so you start continuously driving the system away from it.
Okay, and then it's possible that after the disequilibrium in products has driven some further reactions, that the results of those reactions are molecules which have a lower entropy state. Is this kind of the point?
No entropy, free energy. The thing minimized is free energy. So let's do an experiment, a theoretician experiment. We take a box that is closed, and we wait, it reaches equilibrium. I take a drop of one of the chemicals. Poof, the drop goes in. I have now a disequilibrium. The reactions will evolve in such a way to restore equilibrium. But I'm always going down in G. So just after the drops goes in, I suddenly have a G that is out of equilibrium. And the reaction will decrease it until we get to equilibrium. The closed system is very easy because it always minimizes G.
What's happening in the case of photosynthesis where there's
this is light. So no, it's a good point, of course. And we also did that. Here, we drive our system out of equilibrium with chemostats by exchanging species. Photosynthesis, you're exchanging light. The photons that come from the sun, they are at 5,000 K. Your solution is at in K to, how much is it?
310 or something.
25 degrees Celsius is in K, 300K. So at 300K. So you have a gradient of temperature, and that is actually what is driving your system. So you can also drive, because in that case, chemistry is coupled to light, and light is not at equilibrium with the solution. And that's the mechanism by which you drive your system out of equilibrium. So this is actually an important remark. You can also drive it by light.
How complicated do you think it would be to frame the replication of DNA in this sort of framework?
No?
Biochemistry, then of course depends on what level of detail, but each monomer incorporation is not an elementary reaction. There are a number of steps, and these steps are reasonably well characterized, so people know the rates and so on to a certain extent. But yeah, I think this can be described with this formalism, sure.
In the interest of time, may I suggest that we stop the questions here for now and go for lunch and discuss in the break. Thank you, Massimiliano.